Anybody heard of it before?
Parasite, but it doesn't hurt what it's living off of.
Yes, it's not a parasite, okay? But it does live on things, okay? And the reason why it's not a parasite, it doesn't invade the trees or the branches. And epi means upon, so it's a plant that just lives on top of other plants. And bromeliads and orchids are also epiphytes. They do not have roots. They get their moisture from the humidity in the air. So this is almost the northern boundary that you're going to see Spanish moss, because it really gets too cold and not be in. Angiosperm is a group. Phylum. Ooh, how quickly we forget.
Look at the biology to make sure in your mind.
I know, I'm looking at it. I'm trying to see it.
Anthophyta or Anthopoda?
Yeah, that's what it is.
Yeah, yes, okay.
Oh, yeah.
Uh-huh. And actually, the Spanish moss is a relative of the pineapple family. Okay. Now, if we take a look right behind you, up here, we're going to see a holly tree right here with berries. Okay. And if we take a look at some of the other holly trees, maybe that one or one up there, we don't see any berries. Okay? Why does this one have berries and that one over there doesn't?
Male,
or that one doesn't have them?
Male and female?
Yes, so this is male or female?
Female.
Female. Why is it female?
Because it is berries or fruit.
Yep, it makes fruit. Females make fruit. What do the male trees make?
Pollen.
Pollen. Okay? So if you have a holly tree in your yard that's never had berries, there's nothing wrong with it. You just happen to have a male holly tree. Okay? If we take a look down here, we're also going to see some ground cover with little red berries down here. Part of what we call wintergreen. Okay?
Is that really wintergreen?
I think it is. It's either that or I'll come up with another name. Okay. Let me make up a name. Okay, now. All right. Okay. Most of these are edible. The birds like to eat them. Okay. We're not going to ask you to eat any of this. Okay. We don't want to get anybody that gets sick from these things.
What is Chicago doing today? You're that old.
Right here. All right, but straight down this way, you can see this cordgrass, Spartina and Juncus marsh. And if you take a look at where the trees start, it's anywhere between 10 to 12 to 20 some odd feet back from the marsh area. Now, the reason is why these trees are back farther from the marsh is this Long Creek. Now, if we keep following Long Creek out, it goes up to the Lynnhaven River. If we follow the Lynnhaven River, it goes over to the Chesapeake Bay. And the Chesapeake Bay, of course, comes into the Atlantic Ocean. So we have salt water from the Atlantic Ocean. Chesapeake Bay is salt water. Lynnhaven River is kind of a mixture of brackish between salt and fresh. And the farther away from the ocean and the bay it gets, it becomes much more fresh. Now, Long Creek is coming off of the Lynnhaven River. There is salt here in the Long Creek. So the trees do not like salt water whatsoever. But the grasses, the cordgrass, Spartina and Juncus, they are adapted to being able to live in this very, very salty environment. So what we're going to see in our data, we're not going to collect data here today. We're going to the swamp to collect data. But you're going to see that the salinity of the water here at Long Creek is going to be much more saltier than it is at our swamp. So that means that we're going to see very different kinds of plants from the salt marsh to where we are in the swamp. Okay? Now, also, we have, if you take a look, because there's not a lot of trees where the cordgrass is, okay? It's very sunny, very open, lots of sunlight here. So it's much different conditions in the summer, much kind of warmer temperatures in here because of the sun. So we're going to see some real differences in our physical environment here versus the physical environment in the swamp. Now, right in front of you happens to be a pine tree. These two big ones, or three big ones that are right in front of you, these pine trees right here. This is shortleaf pine, and if you take a look at it, there's just all sorts of pine cones all over the place on it, okay? And if you take a look at your feet, there's some pine needles down there, and there's some mixture of these pine needles. So we're going to get some that are long, okay? But we're also going to see some that are very short, okay? Some of these little short leaf ones versus the long ones. So if we were to take a long one and compare it with a short one, okay? This is probably from that pine, or probably that pine right there. This is our most common pine right over here that doesn't have as many pine cones. It's called loblolly pine, Pinus taeta, okay? And this is shortleaf. If we take a look at how many needles are in one of these little bundles, we call these bundles fascicles, there's 3, and the needles are twisted. We take a look at the shortleaf pine, 2 needles. Okay, so most of the pine trees, sometimes you can look at the bark, sometimes the barks are different. Now in this case, the barks look very similar between that loblolly and these 3 short pines. But you can really tell with their needles and their cones. So if we take a look at some of the shortleaf pine, little teeny cones, little tiny teeny ones. Okay, the loblolly ones are much bigger. Okay, now these even have what we call little prickles on them, and the loblolly cones, yes, here's one. Okay, these loblolly ones, really sharp little prickles on them. Now this happens to be a female cone, and it got knocked off the tree before it was really ready to be released. And then these are normally the green ones you see after the big storms sometimes. And the scales are open just a little bit to allow the pollen to get in. And then they close up, and for a year, the sperm travel to the ovule, to the egg, and start development. So it takes almost 2 years for a pine seed to go from egg and sperm to seed. Okay, when they're ready to be released, then they dry up and their scales open up like this to allow the seeds to come off. Okay? All right? So this is a little bit of the Spartina salt marsh. Unfortunately, you know, with state parks and everything, they try to designate certain areas for conservation only. So there will be no building in this state park. But across Long Creek, these houses have been there for 40 or 50 years, right? And a lot of times with conservation, you have to balance conserving land and then allowing humans to use land. So for the grass and trees over here, they have to deal with the fact that there are humans that are living over there that are basically, you know, not really trying to, but they do pollute the water. Okay, there's boats in the water. That means chemicals, oils, gases, and those kinds of things. Number 2, there's pools over there. So in some cases, some of the pools, if they want to drain it, some of that water gets into the Long Creek, so there's chlorine or salt in there. Also, there's fertilizer on those yards that runs off, and so now you have nitrates and phosphorus coming into Long Creek, which could possibly do some eutrophication, in other words, kind of pollute the rivers. But most of the time, you know, humans are trying to do the best they can without making too much of a mess. But in conservation areas, it's very, very difficult to find pristine areas. So on the edges, these conservation areas have to deal with human impact on them. Okay? All right, so now we're going to go back a little ways. We're going to take a little bit of an alternate route. So back downhill a little bit. To take a look at one of the swamp areas. This is not the swamp where we're going to be, okay? But this is one of the swamp areas that we see throughout this area, wherever there's like a little depression. Now, there are some areas we call swamps, and there's some areas we call wetlands, and it has to do with how much the soils have water in them or on them. So wetland soils have water, they're kind of like waterlogged soils for most of the year. And there's also some soil testing that can be done with wetland areas to find out where the wetlands are and how far back from particular areas they are and how extensive they're found. And there's a lot of jobs in the private sector for environmental kind of assessments for those. So anytime there's some development being done, a lot of these wetland scientists will come in and take a look at the areas and do some soil testing and do inventories of plants, because there's certain plants that live in wetlands. The U.S. Army Corps of Engineers is the federal agency that is the oversight for wetlands protection. So if you buy a house and you have a river, a stream, or a marsh behind you, if you want to build a dock or a bulkhead or anything else like that that's going to be in the wetland area, you have to get permits from the Army Corps of Engineers. And but what we have here is not really what we would call wetlands. This is going to be what we call a swamp. So a swamp is different than a wetland because swamps have standing water most of the year. And that means that it's above the soil line and it is out here so that you can see it like this, okay? Now, what we have here is a cypress, bald cypress swamp. There's probably some tupelo in here. I don't see it right off the bat. But if you take a look straight up and you see these trees with these kind of orangey, rusty needles, okay? This is what they look like. All right. Now, these bald cypresses, Taxodium distinctus, okay, is a gymnosperm. Okay. Now, what do you know about gymnosperms? Most of the time, they are
non-flowering.
okay, right? Naked seeds. Okay. What's the difference between that pine tree and that bald cypress? What does it look like? Color. Color. One is evergreen. This one is not going to be evergreen. This is actually one of the very few deciduous gymnosperms. Okay? Deciduous means they lose their leaves. So right now, these leaves are needle-like right here. Okay? It is in the process of the leaves or the needles are dying. That's why they're turning brown. And they will eventually fall off and then they will get new ones in the spring. So it's a very unusual tree that it loses its leaves in the winter. Now, if we take a look at the bottom of our bald cypress, you can see that the trunk comes down and then it kind of gets bigger towards the bottom. We call that buttressing. So it kind of swells up and forms kind of a bigger bottom to it. Now, these trees are very, very tall, okay? And they're in wet, muddy soil. So the wet, muddy soil is not as stable as dry soil is. So by building kind of a bigger base to it by this buttressing, it helps it stabilize itself. Now the other thing that it does to help stabilize itself is you see these little short knees that are here. These actually are called pneumatophores, okay? And I don't have a chalkboard to draw it, and I can't tell you the truth. I'm not really good at spelling off the top of my head. It's P-N-E-U, pneumatophores, M-A-T-A-P-H-O-R-E-S, something in that category, okay? But we call them cypress knees. Much easier to spell, all right? Now, what these pneumatophores or cypress knees are, there's two thoughts, okay? That they come from the roots of these big trees, and they come up into the air so that they can get oxygen. Now, most roots, okay, are in the soil. There's oxygen in the soil, and there's dissolved oxygen in the water. Now, if we take a look at the swamp, and the ones that we're going to, there's not a lot of dissolved oxygen in this swamp. And the reason is that there is a tremendous number of bacteria in here that are doing decomposition. And basically decomposition is aerobic respiration. They're breaking down materials, and they're going to use oxygen to break things down, and they're going to give off carbon dioxide. So anything that has oxygen in the water, the bacteria is going to take up. So that means that the roots are not going to get a whole bunch of oxygen because there's not a lot of oxygen right here. So one thought is that these knees come up to get oxygen. Number two, it's also thought that these knees are kind of like tent stakes. They put them out, so these help to secure the tree. So it has a whole bunch of places that it can stabilize itself. So this is a very good representation of a cypress swamp right here. Very, very pretty. Okay. All right, questions? All right, we're going to go back up the path and keep on going that way. And it's also, its common name is bayberry. What else is it called? I don't remember. But it makes bayberry. Those little round, kind of silvery looking gray-green berries. And if you were to take one of these leaves, okay, and kind of smash it up, and snip it, it's got a real nice little odor to it. But it's one of the wetland plants that you will see in many areas that is very common around here. It grows pretty fast, and it makes a nice screening for a lot of places. So you'll see a lot of people with these in their yards and things. I'll just pass this around if you want to take a look at it. You can kind of tear up the leaves. Okay. All right, so now we're going to take a look at some of the abiotic characteristics of this swamp area here. So we're going to look at air temperature, we're going to look at water temperature, we're going to test the water for certain components like phosphorus, nitrogen, silica, we'll test the pH, okay? And we'll take a look at light intensity, and we're going to take a look at salinity. Now, if you take a look at this area right here, we've got a little hilly area around here. It's a little hilly over there. You can see kind of the tops of the hills over there. and it's got a little bit of elevation on this side. So this is kind of like a depression area right here. Right? This water has no connections to rivers or streams or the Chesapeake Bay or anything else. So do you think, and the water is only going to come from rain and groundwater. So do you think this area is going to have more salinity or less salinity than salt marsh? Less. Okay? Now, this area here also, as we said when we stopped off at the last place, this has got bacteria in it. And if you kind of take a look at kind of the top of the water, it looks like it's got a little bit of a film on it. Okay? That's the bacteria. Okay? And it's not going to make you sick or anything. You wouldn't want to drink this water, but they're doing decomposition in here. And sometimes if you take a look at the water, you can actually see these little gas bubbles coming up out of the water. Okay? Now, that could be from turtles, okay? Or most likely it's coming from decomposition. What gas is coming up from decomposition?
CO2.
CO2. Okay? So oxygen's being taken out of the water, CO2 is being made. So if we're looking at the decomposition going on, there's a lot of leaves, there's a lot of needles in here from the bald cypress. Take a look up, you can see some of the bald cypress leaves that are turning colors here. This water is going to be very, very acidic compared to Long Creek, okay? Because there's much more decomposition going on in here than there is there. There's a lot of tannins in the water that come from the plant materials. So when we pull the water up, it's going to be a really pretty color, okay? So here we only get rain and groundwater coming in. In the summer, sometimes the water levels do get pretty low, and occasionally if we have a summer that doesn't have a lot of rain, it will actually even dry out for a period of time. But for the most part, all through the year, this has got a lot of standing water in it. Okay, so what we're going to do first is we're going to collect some water. Okay. Now, I have some jars, and we're going to take chemically treated water out of the park. Okay. Any water that we put chemicals in, we do not want to put those chemicals back into the natural water source. Okay, so you're going to empty your testing materials into that bottle right there. Who's got the backpack? Okay.
Is that the difference between the swamp and marsh, where the water comes from?
Marshes are kind of, it's an area that we're going to find around the edges of creeks and streams. Okay, so in most cases with swamps, they tend to be kind of not really, in many cases, connected with a flowing system. There are some that do, but for the most part, they're kind of isolated. All right, so we are going to test. I've got sulfide. I'm going to work in groups of 3 or 4, so I'm just going to hand them out. Okay, sulfide, ammonia, pH. Okay, what you got? Nitrate. Handout that you have, okay, it's going to talk about,
It's also a concentration. It's a small concentration, so it's not pure. What you get is pure.
You got the right one? All right, turn that up.
What you're talking about is parts per million, parts per thousand.
All right, we're gonna look at salinity. Okay. Now, just to give you an example of salinity in the ocean, salinity in the ocean is 35 parts per thousand. Okay, 35 parts per thousand. Long Creek has a connection to the Lynnhaven River that's got a connection to the Chesapeake Bay that's got a connection to the ocean. Okay, this has no connection to the ocean. So we're probably, as we therefore are going to expect low salinity. Okay? Alright, so I'll give that. Alright. Okay, so I'm going to come by and I'm going to put some drops of water on your refractometer. Huh? Yeah. Just a couple of drops, and then find a light source and take a look at it.
Smush it down?
Yep. There you go.
The sun is like right here. You want to stay right here?
Well, here, just take a look at it.
Now I want everybody to look through the refractometers. Even if you don't have one with you right now, share.
Do you see anything?
Glasses might make it look different.
You cannot use it when it's dark over there. You got to come over in the light. Make sure that you look at it in the light, in the sunlight.
I'm guessing 6. The white line represents salinity. Watch my paper.
I don't know. I can't see with my
Do you see anything?
I think the markers on the left